All right, so in this video, we are in Chapter 8, but this video is actually going to address Chapter 8.1 all the way to 8.3, and we're actually just going to be discussing very specific sections of information in 8.1 to 8.3. So in 8.1, we're going to talk about classification by carbon and energy sources. So classification by carbon and energy sources. All right, so classification, of course, meaning classification of our prokaryotic organisms or organisms in general by carbon and energy source. So first we'll talk about classification by carbon source. So when we're talking about by carbon source, we can use prefixes. And the two prefixes that we are going to use are auto and hetero. So auto, the prefix auto, means self, right? So auto meaning self. And when we're talking about self, what that means is that they get their carbon from inorganic sources, and then they're going to utilize those inorganic sources and turn them into organic compounds. So when an organism is classified as auto regarding their carbon source, that means they get their carbon from simple inorganic sources. So when we're talking about simple inorganic sources, what we're talking about is something like carbon dioxide, for example. So taking that carbon dioxide and turning it into an organic compound. So for an example is carbon dioxide. So then the second one is hetero. And when we're talking about hetero, that means other. And so then in this case, what we're talking about is getting their carbon from complex nutrients from other organisms. And namely, that comes from autotrophs. So get carbon from complex nutrients of other organisms. So hetero meaning other, they get their complex molecules from others, and auto meaning self. So they get it from other things in the environment, but they're inorganic carbon sources, so things like carbon dioxide. Versus when we talk about hetero, what we're talking about are things that are getting them from autotrophs, so things like humans, for example, where we get our organic carbon from plants, for example. So when we say auto and hetero, these are the prefixes that are attached to troph. And troph is a root word, and that root word means nourishment, or how they get nourishment. So when we say autotroph, we're saying troph meaning gets its nourishment from, and then auto self. So an autotroph then would be an organism that converts CO2 to an organic carbon. So it's changing it to an organic carbon source from an inorganic carbon source. And then, as we just mentioned, our heterotroph then, troph meaning nourishment, and hetero meaning from others, means organisms that rely on complex nutrients, often from autotrophs. So some examples, again, a heterotroph would be like humans, right? So we rely on getting our nutrients from autotrophs, meaning the plants that we eat. And the plants are autotrophs, meaning that they get it from carbon dioxide. So plants get carbon dioxide from the environment, they convert that into glucose, for example. Then we eat the plants and we get the glucose, which is a complex nutrient, and then we're able to use our carbon that way. So this is how we're identifying based on carbon source, using either auto or hetero. So then the other piece of this is classification by energy source. And when we talk about energy source, what we're talking about is the two different prefix terms called photo and chemo. So again, we're going to use troph as in nourishment, so getting their energy nourishment or their energy source. So the first one is phototroph. So if you can imagine what phototroph means, it means an organism that gets its energy. So that gets energy from light. So photo meaning light, kind of like photosynthesis is synthesizing from light, photosynthesis. This is phototroph, meaning it gets its nourishment, its energy from light. Now, just a clarification, all energy comes from a transfer of electrons. So energy comes from a transfer of electrons. And what that means is that all of its energy is going to come from some sort of chemical process that's going to be transferring electrons to gain energy. But the differences that we have here is the different sources. So really, we have a lot of the same processes happening within these organisms because we're transferring electrons to get this energy, but the electrons are coming from different places. So in the case of a phototroph, the electrons causing the transfer of electrons is coming from light, and then our chemotrophs. So chemotroph, as you can maybe guess here, is organisms that get their energy from chemical bonds. So chemical, chemo, and then light is photo. So in this case, plants, for example, would be phototrophs, right? We all know that plants, when we think of general plants, bushes and trees and things, need to get their energy from light. So they have to be exposed to the sun so that they get energy from light. If they don't have that source, then they don't get energy and they die. So they get their energy from light, and then they are able to go through the process of making different things inside of it, so namely glucose in this case, in this example. And then as a chemotroph, us humans, we are chemotrophs. So we get chemicals from others, meaning if we eat plants, then we get the glucose, for example, and we get our energy from breaking down that glucose molecule. So they are very similar because when we're talking about autotrophs, we're talking about carbon dioxide and then making something like glucose. And a heterotroph, taking that glucose to get carbon, but we're just talking about getting carbon when we're talking about carbon source. Then when we're talking about energy, we are also talking about taking that glucose molecule, but instead of just how we get the carbon from it, we're actually getting energy from it also, being a chemotroph. So then what we can do is we can put these terms together. So when we put these terms together, there's actually a table in the chapter that does this really nicely also, but I'll go through the terms here. So we have chemo, autotroph. So if we're combining these terms, right, chemo meaning the energy source is from our chemical bonds, auto meaning they're getting it from inorganic sources, the carbon from inorganic sources. So we can say chemical bonds, we'll just make the chart or the table. So energy source and then carbon source. So chemo, autotroph, getting our energy from chemical bonds, and then our carbon source is from inorganic molecules, so something like carbon dioxide, for example. So other examples of organisms that would do this would be our hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. So I'm getting this from the table so you can see that. So basically what we're talking about are different types of bacteria that are able to oxidize things like hydrogen, sulfur. So again, this is due to the inorganic carbon source, so that are able to oxidize iron, oxidize nitrogen, oxidize sulfur. So chemoautotrophs. The next one is our chemoheterotroph, right? So just combining these terms, again, chemo, meaning they get their energy from chemical bonds. Then hetero, meaning that they get their carbon from complex nutrients, so from organic compounds. So when we're talking about a chemoheterotroph, this is what we're talking about ourselves, for example. So chemoheterotrophs, we're going to kind of connect this up here. So we're talking about all animals, fungi, some protozoa, some other bacteria too. But I think that when we think of chemoheterotroph, it's easiest to think of ourselves. Then we can switch over to our other energy source, right, and combine it with the previous carbon terms. So we have our photoautotroph. So again, our energy source in this case would be light because we have the photo at the beginning, and then auto, meaning our carbon source is from inorganic compounds. So when we're talking about photoautotrophs, what we're talking about, a good example would be plants. Right, so plants get their energy from light, and then they get their carbon source from inorganic compounds, like carbon dioxide, for example. But we can also think of things like algae, which is typical. You can kind of think about that. Cyanobacteria. All of these things have a lot of things in common here. What we're talking about are things that are photosynthetic, right? Because they have to get their energy from light. So we're thinking these things that do photosynthesis, right? So then also we have our green and purple sulfur bacteria. Then our last term would be combining photo with hetero. So we have our photoheterotrophs. When we talk about our photoheterotrophs, again, from light is our energy source given the photo, and then hetero meaning from organic compounds here. And then the example here is green and purple non-sulfur bacteria and heliobacteria. So remember, there's a difference between green and purple sulfur bacteria, sulfur meaning getting their carbon source from the sulfur compounds, versus green and purple non-sulfur bacteria, meaning those that get it from organic compounds, not from sulfur compounds. Which is why, of course, we see that difference between our photoautotroph and photoheterotroph here. So up here would be the green and purple sulfur bacteria here. All right, so that kind of sums up 8.1, or the section in 8.1 that you should be familiar with. That might be new information regarding our microorganisms. So now we're going to move into the section in 8.2 that you should be familiar with. That's a little bit different than what you may have seen in other classes. So 8.2, and in 8.2 what we're talking about are other glycolytic pathways. So in other glycolytic pathways, the reason we're going to talk about this is because oftentimes when we talk about glycolysis, what we're talking about in previous classes is the EMP pathway. So glycolysis that you are likely familiar with is actually called the EMP pathway. And the EMP pathway is what most students are taught when they're talking about biology. This is where we have our energy investment phase, and then we have our harvesting phase. So if you remember our energy investment phase, this is where we put in 2 ATP for the first part of glycolysis, and then we have our harvesting phase. And in the harvesting phase, we get out 4 ATP, which gives us our net 2 ATP. And remember that in the energy investment phase, what we're doing is we're putting glucose into the system, and then we end up with our G3P molecules times 2. And then, that's a 3, and then we end up at the end with 2 pyruvate molecules. So, just recalling that's our typical EMP pathway, which is our typical glycolysis pathway. And so when we talk about other glycolytic pathways, what we're going to talk about are 2 different ones. The first one is the ED pathway, and the second one is the PPP. So the ED pathway is named this because it is the last names of the 2 people that discovered it. So it is the Entner-Dudoroff pathway. So ED, Entner-Dudoroff. So when we're talking about the ED pathway, what we're really talking about here is just an alternative. So there are some bacteria. The one that they talk about in the text is Pseudomonas aeruginosa. So Pseudomonas aeruginosa is one that only uses the ED pathway. But some bacteria, and they give the example of E. coli, so only ED. And then they give the example of E. coli. E. coli can switch. They can use either the EMP pathway, the typical glycolysis, or they can use the ED pathway. And then this is just going to depend on the circumstances for the bacteria. It's going to depend on what they have around them. It's going to depend on what they need. Because when we're talking about the ED pathway, What we're talking about is taking our carbon source and instead of it being glucose and then glucose being broken down to G3P, that's then being broken down into pyruvate, in the ED pathway, we're going to take glucose and then we are going to go through a series of reactions that you don't need to know the details of. But what we end up with is ethanol, and it only gains 1 ATP. So in this case, if we take a look at our regular EMP pathway, we're starting with glucose, the same thing. We end up with our pyruvate at the end, but we're netting 2 ATP. If we use the ED pathway, we also start off with glucose, so we need a glucose source if we're talking about the ED pathway. But in this case, we end up with ethanol and only 1 ATP. So we see that difference here. So again, it depends on the circumstances. If the organism is looking to make some ethanol, particularly, it would just be using it or have it as a byproduct. But it depends on what is required at the time and what is available. So then the third type of pathway is the PPP, or the pentose phosphate pathway. So this is the third type of glycolytic pathway. And this one is different because there are a lot of different intermediates that are important. There's evidence that shows that this is the oldest glycolytic pathway. So we can say the most ancient is how the text puts it, an ancient universal glycolytic pathway. And that this may be the oldest or most ancient pathway because of these intermediates. And so these intermediates that I'm talking about are used to make nucleic acids. And amino acids. So the reason that they think this is the oldest pathway is because of the ability to not only make nucleic acids based on this process, but also to make amino acids. So if an organism is choosing, if it has the ability to choose between these three different pathways, if we're in a situation, or the bacteria is in a situation where it just needs a lot of ATP, it doesn't necessarily need to make nucleic acids. Maybe it's not going through division right now, so it doesn't need to go through replication. Or making RNA, or maybe it doesn't need to make proteins right now. Right now it's just focusing on making energy. So maybe the EMP pathway is the best way to go for that organism because it gets the most ATP out of it. Conversely, maybe then it's in a situation where it needs to make some nucleic acids as well as some proteins and get some energy. So then maybe it's going to go through the PPP, or the pentose phosphate pathway. So in this case, it's still going to be utilizing glucose, but it's also going to be generating NADPH. So the energy is coming from NADPH in this case, rather than ATP, but it is getting energy because it's producing NADPH that can go into the electron transport chain and chemiosmosis. But then it's also going to be producing those intermediates. So those can be used to make nucleotides and amino acids. All right, so the third piece that we're going to talk about here, so 8.3. In 8.3, what we're going to talk about is the electron transport system. Or the ETS. So you should be familiar with the ETS, the electron transport system, as far as studying things in previous courses. So we are just going to talk about some of the differences between the bacteria that we're talking about or the microorganisms that we're going to be addressing in this class and the typical electron transport system. So the electron transport system, if you recall, is typically when we're talking about eukaryotic organisms inside the mitochondria, but obviously in bacteria that's not the case because there are no mitochondria. But we have our series of membrane proteins, and it's going to utilize the electrons from NADH and FADH2, and we're going to be passing those electrons along. So when we're talking about the electron transport chain, we're talking about this is the last piece of cellular respiration. So I can probably write this down. So the last component last component of cellular respiration. So at this point, just as a refresher, we've gone through some form of glycolysis. Then we kind of skipped through it, but then also remember that there's the Krebs cycle second. There weren't any differences in the Krebs cycle that we needed to discuss between what you probably have already studied in a previous course and what we're going to be looking at and utilizing in this course for identification of microorganisms. So we didn't address anything specific in the Krebs cycle. But do recall that the Krebs cycle does occur. So we do have the Krebs cycle in these different types of microorganisms. We spoke about glycolysis, the 3 different types of glycolysis, so the EMP, the ED, and the PPP. So we have those different types of glycolysis that we'll see as we're moving forward in the course and determining the differences between different organisms. Then all of these organisms are going to go through the Krebs cycle, which is pretty similar across the board here. And then this last piece is the electron transport system. And then also there's chemiosmosis. And proton motive force, things like that, that we're not going to talk about specifically because, again, it's similar to what you have already studied in previous courses. But here we are in the ETS, that last component of cellular respiration, so producing energy. Excuse me. So then again, the ETS is a series of membrane-bound proteins. So a series of, we'll say, membrane-associated proteins and mobile electron carriers. All right, so typical of what you've studied previously, we have the membrane, we have these proteins or protein complexes, and then we have these other mobile electron carriers. And then we have a series of chemical reactions in which the electrons are passed from FADH or FADH2 and NADH, and then they're passed to these electron carriers with these increasing positive redox potentials, and then it gets all the way to the end of the electron transport system, and that's where we're going to see a lot of these differences we're talking about. So, a series of chemical reactions in which electrons are passed from, remember it's NADH and FADH2 are electron carriers, to electron carriers. So these mobile electron carriers, and then these protein complexes with increasing positive redox potential. So meaning positive redox potential, meaning that the electrons are going to be moved from something that's a little bit more positive and a little bit more positive. It's going to be moved along these series of protein complexes and these mobile electron carriers. And then we have different classes. So the text does mention the different classes of these electron carriers. So we have cytochromes, we have flavoproteins, we have iron, sulfur proteins, and quinones. So the reason that we're mentioning these 4 different classes is because as we move through future chapters in the semester, we're going to be taking a look at differences between different types of bacteria. And so some of these classes are going to be in consideration when we're doing different tests. And we'll talk about that in just a moment here, even specifically with when we're talking about aerobic respiration and we're talking about the different types of cytochrome oxidases. These cytochromes, which are going to allow us to tell the difference between different organisms. So let's, I guess, jump into that right now. So we have aerobic respiration. Right, and so aerobic respiration, meaning contains oxygen. So when we have aerobic respiration, our final electron acceptor is oxygen. So, of course, being aerobic respiration, that's what this means, is that our final electron acceptor at the end of the ETS is oxygen. So when that electron gets toward the end, then what we have is that our oxygen is going to become reduced to water. So it changes to the H2O. That's why we have a lot of water is the byproduct here. And then that's what ends the electron transport system there, is that electron is moved over there. That energy is going to reduce oxygen to water. So the electron carrier that does this is our cytochrome oxidase. So oxidase. And that is what's going to differ between different bacterial types. So this is something that we can use for diagnosis. So when we have different types of cytochrome oxidases, we can say that it varies between closely related bacteria. So when we are trying to identify something, we can do these kind of growth tests, meaning these larger tests to separate between major classes or major genus or major species. But then when we get down to kind of the smaller differences between things, we can use things like taking a look at the different types of cytochrome oxidases. So we can see that we can then determine what type of cytochrome oxidase it has, and then we have a better idea of what organism we're talking about. So some of the examples that are in the text are Pseudomonas aeruginosa and Vibrio cholera. They both use the cytochrome C oxidase, so they end up being positive when we do an oxidase test. But then on the other hand, we have E. coli, and that uses different oxidases. So we have the different types of cytochrome oxidases can help us determine what we are looking at. Excuse me. So oftentimes organisms, if they can, they'll do aerobic respiration. And that's because we get so much energy from aerobic respiration. Remember from previous courses, you get about 36 to 38 ATP from aerobic respiration. So that's a really, really good option for getting energy. But there are often reasons why organisms cannot do aerobic respiration. So when we can't do aerobic respiration, an example would be, there are 3 different examples that your text talks about. First of all, the cell actually lacking the genes to encode for this appropriate cytochrome oxidase. So the cytochrome oxidase is what is transferring the electrons to the oxygen to make that H2O. If the gene or if the organism just doesn't have the genetic material to code for a cytochrome oxidase, then it can't do aerobic respiration. So that's number 1. So the cell codes, or rather, the cell lacks the code. So the cell lacks the code for cytochrome oxidase. Or other transfer electron at the end of the ETS. So it could lack something else also, but typically what we're talking about is lacking the code for the cytochrome oxidase at the end there. The second reason would be that the cell lacks the genes to combat the oxygen radicals that are produced. So aerobic respiration actually is quite dangerous. It produces a lot of very dangerous byproducts. And these are oxygen radicals. A lot of times people talk about them kind of generally as free radicals. So these dangerous oxygen radicals are produced during aerobic respiration. So that means in us as well. Now we have a lot of different enzymes and things to try to combat these dangerous side effects or these dangerous byproducts of aerobic respiration. But if a certain prokaryotic organism or microorganism lacks the genes that encode for these enzymes, then they're not going to be able to go through aerobic respiration, because if they did, then just the production of these oxygen radicals are going to kill the organism if they don't have the ability to get rid of these dangerous byproducts. So it could have the code in its DNA for the cytochrome oxidases. However, it still wouldn't be able to do it because the cell, in the second option here, the cell lacks the genes to combat the oxygen radicals produced during aerobic respiration. So some of these oxygen radicals are like hydrogen peroxide is produced. Remember, that's why we have something called peroxisomes, which are little vesicles that are filled with enzymes to get rid of hydrogen peroxide because it is dangerous. We also have something called superoxide, which is another oxygen radical that we need to get rid of utilizing different enzymes. So we have the cell's inability to produce any of these enzymes to get rid of these different types of oxygen radicals. But then third, the cell may lack a sufficient amount of oxygen to carry out the aerobic respiration process. So even if the cell does encode for cytochrome oxidase, even if it does encode for the enzymes that can help combat the oxygen radicals, it may just lack oxygen to carry out aerobic respiration. So it finds itself in an environment that has very little or no oxygen at all. So it just can't produce or cannot go through, aerobic respiration. So the cell lacks a sufficient amount of oxygen. So there just isn't enough oxygen around in the environment. So then what's an organism to do? So an organism is then going to go through anaerobic respiration as an alternative. So again, the first or primary or the best option is aerobic respiration. So for organisms, we get the most energy out of aerobic respiration. But if a cell finds itself in either of these three situations, so for example, if it doesn't have cytochrome oxidase, doesn't have the genes to combat the oxygen radicals, or there just isn't oxygen in the environment, then it would like to have an alternative. Now, that doesn't mean that all microorganisms do have an alternative to aerobic respiration, because they don't. Some of them can only undergo aerobic respiration. Those are called obligate anaerobe or obligate aerobes, and we'll talk about those more in, I believe, chapter 9. But some organisms are what are called facultative. A facultative organism, I'll put it that here, facultative organism is when the organism can switch between aerobic respiration to anaerobic respiration and back and forth. So can switch between aerobic respiration and anaerobic respiration. And this is important for bacteria. It's a really good thing for some bacteria, so that if they have the ability to code for all of the different things to go through aerobic respiration, they will if oxygen is present, that third bullet point here. If oxygen is present, they'll do aerobic respiration and get the 36 to 38 ATP and have lots of oxygen, or rather lots of energy. If, however, they find themselves in a situation where there's not very much oxygen, then they can switch over to anaerobic respiration. So they can still be making energy, but they just do it in lower amounts, which we'll see here. So in anaerobic respiration, instead of using the cytochrome oxidase, of course, because this is an alternative to that, instead it's going to use inorganic molecules instead of oxygen at the end as the final electron acceptor. So if we don't have that cytochrome oxidase in order to transfer to oxygen, or if we don't have oxygen, then it's going to use an alternative inorganic molecule. So it uses an inorganic molecule instead of oxygen as the final electron acceptor in the ETS. So this is important because what we're talking about is the difference between aerobic respiration and anaerobic respiration. And simply, if we put it very simply, the difference between aerobic and anaerobic is that final electron acceptor. In the case of aerobic respiration, that final electron acceptor is oxygen. In the case of anaerobic respiration, that final electron acceptor is some other inorganic molecule. And it depends on the organism and what's available in the environment as to what that inorganic molecule might be. So in the text, it talks about denitrifiers. And we're going to talk about this in our nitrogen cycle as well. But these types of organisms, these are typically soil bacteria, and in this case they use nitrate and nitrite as their final electron acceptors. So, for example, nitrate and nitrite as electron acceptors, as the final electron acceptor. And so in this case, we're using a nitrate, looks like this, and nitrite looks like this. So we do still have some oxygen here, but then in this case, oftentimes what happens is they can use nitrite instead of nitrate when there is low levels of oxygen still. So some bacteria are able to switch back and forth when there is oxygen to using nitrate and then using nitrite when the oxygen levels have decreased. But in this case, they're going to utilize nitrate or nitrite as the electron acceptors, and then in this case what they'll do is they'll produce nitrogen gas, or N2, which again is important, and we'll talk about it in the nitrogen cycle. But anaerobic respiration uses other different types of inorganic molecules, and we'll talk about some that can include sulfur and things like that as well. In this case, however, in anaerobic respiration, we are producing less ATP. So less ATP is formed in this case than in aerobic respiration. And so that's a downfall of anaerobic respiration, which is why they will go aerobic versus anaerobic. So microbes that use anaerobic respiration still typically have an intact Krebs cycle. I think I said earlier that something along the lines of most or all microorganisms have an intact Krebs cycle. That's not necessarily true. While the ED pathway does have pyruvate as one of its products, it does end up in the ethanol, end up with ethanol at the end. So it may or may not use that pyruvate to go into a Krebs cycle. However, here, microbes that use anaerobic respiration do commonly have the Krebs cycle also. So they can still get the energy from NADH and the FADH2 molecules that then go into this ETS and then end up with something that has an inorganic molecule at the end as the final electron acceptor rather than oxygen in that case. So still smaller gradients, smaller amount of energy in this electron transfer system, so we don't get as much and it's not used if the organism can use aerobic respiration instead. So one thing that I do want to clarify, we're going to move on to chapter 8.4 in another video. However, I want to mention that to be clear between aerobic respiration and anaerobic respiration, because the difference there is just that the molecule at the end, that final electron acceptor, is either oxygen or it's not. So it doesn't say specifically in anaerobic respiration what that molecule is. The example given here was nitrate or nitrite, but it doesn't say what that molecule is. But if there is an electron transport system, something is aerobic if it ends at that oxygen. It's anaerobic if it ends at something that's not the oxygen. And then in chapter 8.4, section 8.4, we're going to talk about something that is different called fermentation.